Okay, what we're looking at is the structural components of the eye and ear. We'll look at a dissection of the eye, but we'll start out with landmarks, structural landmarks. So on the eye, first of all, we have, this is the right eye, and we have, on this structure, we can see the lacrimal gland, which is a structure that's always posterior, which is always superior and lateral. And that superior lateral gland is going to be a good landmark for determining right side or left side. So if it's superior and lateral here on the eye, then this would be the right eye that we're looking at. Another landmark would be the muscular attachment and the pathway of the muscle into the eye. As you can see right here, within the orbit on the medial part, close to the bridge of the nose or the nasal bone, where the maxilla and nasal bone and frontal bone come together, you have the superior oblique attachment, which comes into the top of the eye. You can see on the structure of the eye here, you can see on the structure of the eye, the colored part of the eye, that's the iris, covering that structure and filled with aqueous humor would be the cornea. Deep to that structure we'd have aqueous humor. The pupil is the opening, the iris is the colored part of the eye, and then the white part of the eye in the fibrous layer with the cornea would be the sclera. If we remove this structure and look at the eye a little closer, you can see that all around the outside of the eye, about halfway moving posterior towards the optic nerve in the back, you have muscles that come straight into the side portion of the eye, the top, the bottom, and medial part, and those structures are referred to as the superior, lateral, medial, and inferior rectus muscles. Each of their shortening would cause lifting and movement inward or out of the eye. The only one that's going to rotate the eye, only two muscles that will rotate the eye, would be the superior oblique and the inferior oblique, that attaches again to this medial portion right by the nasal bone and then comes across the top of the eye into that superior attachment there. If we look a little closer and enlarge the model that we're looking at, this is a bigger model of the eye. Again, the lacrimal gland here is the landmark that's used as superior and lateral on the eye to determine that this in fact is the right eye that we're looking at. Notice here on the top we have the attachment of the superior oblique that would come in medially, attach to the bridge of the nose, and then move in and attach to the top of the eye. Here is the superior rectus, the medial rectus on the same side as the superior oblique, the inferior rectus below, and the lateral rectus on the outside. We'd also have on the underside of this of the eye the inferior rectus more visible, but they're both coming in from medial to lateral. As we look at this structure also, we can see the white of the eye, that is the sclera. The sclera and the surface that would be covering the front of the eye is the cornea, and those two together make the fibrous layer or tunic of the eye. As we pull this top portion off, we can see that structures inside. Here's the colored part of the eye right here. That's going to be the iris, and at the center of the iris, being controlled by smooth muscle in that structure, would be the opening, the pupil. The pupil's dilation would be related to smooth muscle contraction and would regulate the quantity of light coming in and hitting the refractive, after it hitting the cornea, hitting the two refractive surfaces of the lens. You can see also inside of the eye that we have a grouping of muscle that surrounds and controls not only the opening to the eye, the pupil, in toward the lens in the posterior segment, but it also controls the suspension ligaments, the suspensory ligaments tension, and therefore the thickness of the lens itself, the second and third refractive surfaces. Just posterior to this ora serrata, or this serrated edge, which contains those ciliary smooth muscles, we have the retina coming all the way back and attaching at the macula surrounding the circumference in the back of the eye here, right there, and that would be the fovea centralis. This is where the image would be refracted to after refracting at bending at three spots. The image would be inverted and smaller and focused at this single point in the back of the eye, and that's where therefore we have a concentration of cones for intense light processing located there. It would go through the layers of the retina to the innermost layer, and those layers are seen right here with each of the four portions of that structure. Here's another view of the same thing. Right, so here is another view of the ora serrata. In that ora serrata, or the serrated edge, we're going to find ciliary bodies or muscles that are attached to suspensory ligaments that are seen here. There would be a lens found right here connected around its circumference by those suspensory ligaments, and as the ciliary bodies shorten or contract, you have tension in the strings that attach to the lens that would be created, and the lens would then thin, creating less refractive surface area. In this, the eye itself is broken up into two major portions. The anterior segment that we see right here, just inside the cornea. Here's the cornea, which is a connective tissue layer. That's the first refractive surface that we see light bending in. Then it would enter this fluid-filled segment or chamber that we have in this anterior segment, and that would contain aqueous humor that's constantly renewed through the canal of Schlemm under hydrostatic pressure. The opening is here through the pupil, and that pupil's diameter would be controlled by the quantity of light and the ciliary muscles. As the refraction occurs, it's going to concentrate the images, focusing right there at the center and through the pupil, and then hit the lens anterior portion, exit the lens into vitreous humor, and then hit, ideally, here at the fovea centralis, in the posterior part of the eye, where the image would be inverted and smaller. As it's focused at this point, you have different layers that make up the retina, and this entire back portion that is filled here would be actually lined with the retina, just posterior to the ora serrata. Then as we move into the layers that go superficial on the eye, we would go from retina, the neural layer, to the pigmented layer of the retina, which is attached close to the choroid layer right here, the vascular layer, number 14, and then that moves out to the white of the eye, all around the outer surface of the eye, which would be the sclera, as we already described. Okay, so again, back to the image. As we focus this image right here at the fovea centralis, we see four basic layers of that retinal or that sensory layer, and the retina itself is made up of these four portions. So if we look right here in this part of the model, the image is going to pass through this portion, through those four layers, and go all the way into this cuboidal layer of cells, number 43, which would be where our photopigment rhodopsin is found. It would be in a retinal would be in a cis bent orientation connected to the various opsin proteins, and as light hits it, we're able to convert or transpose the bent shape of the cis orientation of that retinal, and it straightens out and releases from the opsin, and as those two release, we are able to open sodium gates in the photoreceptor layers of the rods and cones, depending on which opsin is released. As that release occurs, you're going to have a funneling or a summation occurring of each of these depolarization events that will be sent through the bipolar cells, which are less numerous, through the ganglion neurons, and it's on the axonal side, number 37, where we generate the action potential, an all or none response, and reach the threshold. And it's this accumulation of all the axons of that ganglion layer that produce here the actual optic nerve that exits through the optic canal of the sphenoid bone and makes its way after decussating. And with decussation, this occurs on the visual pathway here. So the optic nerve would be here. It exits through the optic canal. It crosses over both eyes, stereoscopic vision, so both eyes are going to send impulses of that image through the optic nerve on either side. Decussation, or crossing over, occurs so that in both parts of the occipital lobe in the brain, we receive the images from both eyes processing. This decussation also is associated here with the attachment of the pituitary gland, and the infundibulum, the pituitary stalk that it attaches to, allows us to create a relationship between visual pathway and the cues that would be related to the release of hormones from the endocrine system. As it continues back into the occipital lobe of the brain in the posterior cranial fossa, it's going to be sent lateral to the geniculate bodies of the thalamus, and that pathway into the occipital lobe for processing is related to memory there on its way back to that portion. An ideal situation for a special sense to create a long-term response, and a response that's fine-tuned and eliminating any non-vital parts to it. Okay, and that would be the eye. You can see the eye is encompassed within fat as well, and each of the muscles here in the right orbit are visible. Is that cool? Is that, you want to stop?
You want to go over the dissection?
Yeah, and as we finished this, we dissected a cow eye. The cow eye, can you zoom in on that?
Yeah.
Okay, the actual cow eye structure is easy to determine. It's encased in fat, so this really looks kind of crude. But what we've done is we removed the front part of the eye, which again was the cornea. So there it is. Looks like a big thick piece of skin from the calluses in the soles of your feet or in your palms of your hands. But we removed the cornea, the first refractive surface. You can see that what we removed contains those ciliary bodies right there, the muscles in the ora serrata, and as we cut this piece off, you can now see into that posterior segment of the eye, and we pushed on the sides of the eye, removing the lens. This is actually the lens from the eye. So that came out through the pupil as we cut it open. We removed the vitreous humor in the posterior segment, and now you can see in this nocturnal animal a sort of fluorescent layer that's underneath this mucous material, and that mucous material represents the actual connected retina and the condyle that surrounds it. So if we pull away this sensory tunic and look underneath, you can see a fluorescent layer just superficial to that, and that would be the choroid layer and what's referred to as the tapetum lucidum, a reflective surface of nocturnal animals, so they can see better at nighttime. Again, what we would look for is this structure on the eye, which would be the lacrimal gland, which is superior and lateral on the eye. So this would have to be the right eye, since that's the lacrimal gland. And we can see that muscles and fat surround the orbit of the eye all throughout. The last thing would be that on the posterior portion of the eye, you can see as you pull this apart an optic nerve right there from the side. That structure here is the optic nerve that is the converging point of all of the ganglion layer axons and the pathway of sensory input to the occipital lobe in the posterior part of the brain. All right, the processing of the equilibrium and balance, static equilibrium and sound are all going to occur within the temporal bone, the petrous portion of the temporal bone, and the inner ear structures. So as the overall pathway of sound would be entering through the pinna and harnessed by the pinna of the ear, which is elastic cartilage connective tissue. We harness that sound and send it through the external auditory or acoustic meatus into and through the external ear till it hits the lateral portion of the tympanic membrane. As it hits the tympanic membrane, it's going to vibrate at the same frequency and amplitude, and it's even going to increase its rate of vibration because we're moving from a gaseous medium into a solid, and so the lost amplification of that sound along the way to the ear is compensated for as it hits the more dense medium of the tympanic membrane. As it vibrates, we then send that vibration through and into the three ossicles, which would be the malleus, the incus, and the stapes. And as it enters those three smallest bones of the body, we associate this middle, on the medial side of the tympanic membrane, this middle ear portion, with a connection of the pharyngeotympanic tube or eustachian tube to the throat portion of the pharynx or airway. And that pharyngeotympanic tube thus is a connection or a possible pathway of infection. If this weren't at a downward angle, in children for example, you may see an accumulation of fluid in the middle ear, an infection setting in here in the eustachian tube, and then that infection possibly spreading into the inner ear structures and causing major problems. So the eustachian tube or pharyngeotympanic tube is at the base of the is at the base of the middle ear, and these ear ossicles that are vibrating and transmitting sound through the middle ear would ultimately be connected to the inner ear structure, which would be the goes malleus incus stapes, and this is the connection of the stapes to the oval window just below or just onto the vestibule of this inner ear trio of structures. If we look at this a little closer in a larger model like we see here, you can see this this oval window. Okay, so this portion that we see here would be the oval window where the stapes was connected of those ear ossicles from the middle ear. We have surrounding this entire shape structure after the connection of the stapes a membrane here, and that membrane is existing on two levels. It's a membrane that is sitting on the bony labyrinth that encompasses this entire inner ear structure. There are three parts to this structure. So as the stapes connects, this is a fluid-filled trio of structures, and the fluid is contained within a bony labyrinth that encompasses all parts of this inner ear structure. The processing that's going to occur here is by modified nervous receptors, neurons, and those neurons are going to be found in the vestibule, which is in the center or the separating point between this snail shell or cochlea and these three canals, which are going to consist essentially of a lateral, an anterior, and a posterior X, Y, and Z plane that are fluid-filled. So this bony labyrinth is encompassing a fluid-filled cavity, and you can see that here. And inside of that fluid-filled bony labyrinth, you see there will be a membranous labyrinth, and inside of that membranous labyrinth we have endolimbs, so two layers of fluid. And if we create at this oval window a vibration, or if the vibration from the middle ear comes in and vibrates this window, we have, as the Doppler effect would show, a radiation of this vibration into all three receptors found here. The receptors we're talking about would be in the vestibule, would be the macula that contains hair cells embedded in an otolithic membrane. That otolithic membrane is surrounded by endolymph in the membranous labyrinth. And as you displace that membrane by moving this fluid, you're moving essentially chalk crystals or calcium carbonate crystals, and as you move them, the mechanical displacement pulls or triggers the action potential in the hair cells that's radiating through the vestibular portion of the nerve that comes out of this area. So if we look at this particular model, okay, we can see the same structures that we're just looking at. This right here would be the stapes of the middle ear. Then it would be connected to the oval window. It's going to be connected specifically on the vestibule that contains the receptor called the macula. The macula's otolithic membrane and otoliths are surrounded by endolymph or fluid within its membranous labyrinth, and as the wave is created in the bony labyrinth, a wave is created internally in the endolymph, and it will trigger these macula structure, and the otolithic membrane moves, triggering a pathway of nervous impulse through this, the vestibulocochlear nerve, cranial nerve number 8, that enters the temporal lobe through the internal acoustic meatus. Now notice there's not only just that middle portion, there's two others. There's the cochlea, which is the processor of sound, and it uses the organ of Corti to do this. We'll look at that in a moment. At the center was the macula, and this is a processor of static equilibrium. In other words, that's head position and linear acceleration. And then we have these three semicircular canals, the anterior, right here, the anterior, the lateral, and the posterior canals, which will allow you to maintain dynamic equilibrium and balance constantly because of fluid movement in each of these planes of existence. Notice that there are three parts of the nerves, the vestibular portions coming from the canals and the vestibule, and the cochlear portion of the vestibular cochlear nerve, both combine and go through that internal acoustic meatus on their way into the temporal lobe after passing by the memory centers, because this is a special sense that we're processing. So what I'm going to do is I'm going to look at with you the processing within the snail shell or cochlea structure, and using the organ of Corti to describe specifically what's happening because what's happening in that location would be equal, with different terminology, to what we have happening with hair cells being displaced mechanically in the vestibule and semicircular canals, the ampulla. The processor, again, in each one of these, in the semicircular canals for equilibrium and balance, in each ampulla would be the crista ampullaris. In the vestibule, we have the macula, and in the cochlea, we would find the organ of Corti. So if we were to blow this structure up and look at a cross-section of it, we have this right here. Okay, so this is a cross-section, or a cross-sectioning of the actual cochlea portion. So that snail shell has been cut lengthwise, or here, and we're looking into the chambers that are contained within it. This is the organ of Corti. We have three portions. So this outer part here is the bony labyrinth, and the fluid that fills the chambers that are the outer portions of this bony labyrinth are called perilymph, or is called perilymph. The perilymph's movement, either whether it's from the eustachian tube and the round window, or if it's come from external and hit the or the oval window through the stapes, either way we're going to create a wave that's going to cause a wave in the endolymph of the cochlear duct, or the scala media. This part up here is called the scala vestibuli. This is the scala media. This would be the vestibular membrane that separates the scala vestibuli from the scala media, or cochlear duct. And then the entire organ of Corti sits on a basilar membrane, and this would be the scala tympani below. Okay, so these three chambers contain fluid that will have a wave that's created in them, as the Doppler effect describes. If you throw a rock into a pool, it'll radiate circularly out from that source of contact, and that Doppler effect is the same thing that's happening here. If we vibrate the oval window or the round window from the eustachian tube connection, we would have a wave created in the fluid that's contained within this bony labyrinth that sends it along the surface of the membranous labyrinth and creates a wave internally in the endolymph of this middle portion, the cochlear duct or the scala media. So either way, wherever that wave is created, either from the scala vestibuli and the oval window, causing movement or a wave in this vestibular membrane, we would have a wave then in this scala media, pushing this structure, the tectorial membrane, down upon these hair cells embedded in this basilar membrane. And as we mechanically make contact with that hair cell that's embedded in the membrane, we trigger the opening of sodium channels, and an action potential results along the cochlear portion of this vestibular cochlear nerve, and sound is processed. Okay, likewise, if we plug our ears and we vibrate our vocal cords, even though there's no external wave of sound coming in, we're creating sound internally. It'll come up from the round window from below, and that round window will trigger a wave in the scala tympani. Okay, and the scala tympani's wave will cause a wave in the basilar membrane and the pushing up of this membrane here, of the organ of Corti that contains hair cells. So you move it from the bottom up into the tectorial membrane, and the contact between the two create the triggering of opening of sodium gates. So an action potential will be generated also on the cochlear nerve.